So I know you're working hard to try to figure out all these organelles and what they do, but I think it's also important to think a little bit about how they work together. So I'm going to tell 3 stories today about 3 possible routes for a protein that's being newly made in the cell. And so there's 3 different ways the gene, the message contained here in the DNA and the nucleus, there's kind of 3 different ways that can be processed in the cell, eventually leading to a functional protein. So the first and the most basic way is that a DNA can get converted into messenger RNA. And I know we don't really know what this is yet, but the information from the DNA is read and copied into a complementary message in this RNA molecule. And that leaves the nucleus. Okay. And in this case, story number 1 I'm going to tell you is just about a protein that has a job out here in the cytoplasm. Okay? And this protein is going to, the mRNA is going to make its way out of the nucleus until it hits a free ribosome. Okay, this little blue ball here is meant to represent a ribosome. Okay, and what will happen here is this message gets read by the ribosome. This is an enzyme that catalyzes the formation of the peptide bond. This is where the MERs actually get joined together. All right, and then out of this ribosome will come the protein that does its job out in the cytoplasm. Okay, so this is the simplest and most direct path between a gene message and a protein. Another possibility is for a protein that has a destination inside a specific organelle. All right, in that case, you're still going to have DNA converted into mRNA. And this will then make its way again out of the nucleus, and it's going to hit a ribosome stuck to, these little blue dots are meant to represent our ribosomes, and this organelle here, the rough endoplasmic reticulum, is studded. The whole surface of this organelle is studded with these ribosomes. Okay, and it's important to note that the nuclear envelope, the membrane of the ER, the membrane of the Golgi apparatus, the membrane of a vesicle, and the membrane of a cell are primarily made of the same components. So what can happen is you get a lot of fusion and melding of one membrane into the next. Okay, I always think it looks like a lava lamp. I don't know if anybody knows what a lava lamp is or does, but it's a lamp with a light bulb that heats up wax, and it goes and forms blobs that melt into each other. So if we're talking about a protein, this is story number 2, we're talking about a protein that is destined for, let's say, for example, a vesicle. All right. This could be something like a lysosome that does the digestion of particles inside a cell. All right, and here what happens is when that messenger RNA hits that ribosome on the rough ER, that protein is made inside the rough endoplasmic reticulum. Okay, so inside that rough ER will be these protein particles. All right, and those are collected and packaged together into a vesicle, and this vesicle carries that protein to the Golgi apparatus. So a couple of important things here happen on this pathway through the endoplasmic reticulum. One is you have quality control. If the protein's not folded properly, it will be eliminated. It will be chewed up and recycled. All right, but if you pass that quality control and you go into the Golgi apparatus, a couple of other things happen. So in the Golgi, you get packaging, tagging, and sorting. All of these taking place here in the Golgi. So the tagging here means that the protein will have sugar residues added to it, and those will help this cell to form little bundles of the protein in, again, these little vesicles. Okay, so that you have little membrane-bound containers that all have the same protein in it. If that protein happens to be a digestive enzyme, that will get sent to a lysosome. Okay. All right, so that's sort of the second route, and this pathway from nuclear envelope with the mRNA to the rough endoplasmic reticulum through the Golgi to a membrane-filled container. This is the endomembrane system. Okay. All right, the third possibility starts out in a very familiar way. It starts out with our DNA. We go to the mRNA. Again, this message goes in and hits a ribosome on the rough ER. Again, you get proteins made inside the rough endoplasmic reticulum. All right, those come together in a container that buds off of the ER. And then again, you go into the Golgi apparatus, and you have the same things happening. You have your sorting, you have your tagging, you have your packaging. So the analogy here is this is like FedEx or UPS, depending on what you use. In the Golgi, you're getting your sugar tags that'll send it. So if you're not destined for an internal organelle, it's possible that you are destined for a membrane-bound vesicle that's eventually going to fuse with the cell membrane, and that protein would have some job outside of the cell. Okay, maybe it's a hormone that has to go act in the blood on a cell that's in a very different tissue in a different part of the body. All right, so the whole process, there's a couple different ways of going through the endomembrane system, or you can just have your protein be made at free ribosomes that are hanging out in the cytosol. Okay, so there's a couple different pathways here that I want you to be aware of, and this is important because it shows how these organelles work together in a cohesive system. All right, I hope that helps. Please make sure to take the quiz that's associated with this video, and good luck.